Good morning. It's November 6th, and this is your daily brief in science. Here's everything you need to know. A new line of research from Osaka Metropolitan University shows adipose-derived stem cells, or ADSCS, can be guided into bone-like spheroids and combined with beta-tricalcium phosphate to boost bone regeneration in a rat model of osteoporotic vertebral fractures. The study reports stronger regeneration and improved vertebral strength, suggesting this ADSC-based approach could help treat difficult fractures and potentially accelerate healing in spinal injuries. Significant activation of bone formation genes was observed, indicating robust engagement of the healing biology in treated subjects. While the findings are promising, researchers emphasize that human safety, effectiveness, and long-term outcomes still require clinical trials. Using autologous fat tissue means patients' own cells are used, reducing immunogenic risk and enabling less invasive outpatient procedures. The study, published in Bone and Joint Research in 2025, also notes practical advantages such as safety and ease of fat extraction, supporting future human applications after further work. ADSCS are easy to collect even from older individuals, and their use involves little bodily stress, pointing to a non-invasive option for bone disease treatment. These cells are multipotent and can be obtained through minimally invasive procedures, presenting fewer risks than bone marrow-derived cells. The researchers describe a tissue engineering approach that combines ADSC spheroids with scaffold materials to provide both biological signals and mechanical support during spinal healing. Preclinical evidence shows ADSCS can contribute to bone regeneration, justifying continued research toward clinical translation. The findings could extend beyond vertebral fractures to other bone defects and non-union fractures, advancing regenerative medicine in osteoporosis care. The work targets elderly patients with osteoporosis-related fractures and outlines a shift toward leveraging the body's own regenerative capacity, reducing reliance on invasive surgery. Publication in Bone and Joint Research on 28 October 2025 signals collaboration across regenerative biology, materials science, and orthopedics as the team moves toward human trials. A new biomaterial vaccine approach targets Staphylococcus aureus infections tied to implanted devices, offering a potential shift in how we guard against device-related infections. Researchers report that biodegradable scaffold vaccines injected near implants attract and stimulate immune cells, producing stronger immune responses in mice than conventional vaccines. The platform uses FCMBL technology to capture pathogen-associated molecular patterns, creating a broad snapshot of bacterial components to enhance and prolong immunity. In tests with MSSA antigens, the biomaterial vaccines protected against subsequent MRSA infections, suggesting broad, off-the-shelf potential for orthopedic procedures. The study appears in the Proceedings of the National Academy of Sciences and is led by David Mooney and teams from SEAS and the Weiss Institute at Harvard, with NIH and Weiss-Harvard support noted in publication credits. The researchers say the platform could safeguard other long implant devices beyond orthopedic implants and may reduce revision surgeries, extended antibiotics, and related complications if proven in humans. In the U.S., hundreds of thousands of knee and hip replacements are performed annually, with infection rates estimated between 2% and 4% for implanted devices. The findings point toward a paradigm shift in vaccine design for device-related infections and suggest clinical integration of biomaterial vaccines as a precaution before implant surgeries. Experimental results show protection against both antibiotic-sensitive and resistant Staphylococcus aureus strains, and the approach hints at personalized vaccines based on patient-specific PAMP signatures to tailor protection before surgery. In a model where devices were implanted and infected after vaccination, bacterial growth on devices was significantly suppressed 5 weeks post-immunization. Beyond orthopedics, the approach could be adapted to protect other implanted devices, reflecting cross-disciplinary collaboration across immunology, bioengineering, and clinical research. Lead researchers incorporated disrupted bacteria-derived antigens into the vaccine and used FCMBL to assemble hundreds of FCMBL-bound PAMP antigens, fostering durable immune activation with diverse T helper cell responses and protective cytokine production. The result is a broader, more sustained immune defense than traditional soluble vaccine formulations, with potential implications for reducing device-related infections across medicine. A new environmental microcompartment genomics approach dramatically increases throughput for single-particle genomics, enabling sequencing of thousands of individual particles from tiny seawater samples at reduced cost and with improved data quality. In a Nature microbiology study, researchers demonstrated the method by sequencing genomes from over 2,000 particles in roughly 300 nanoliters of Gulf of Maine seawater, illustrating substantial gains in speed, scale, and data quality. The Gulf of Maine sample yielded genomes for more than 2,000 particles within 300 nanoliters, highlighting about an order of magnitude boost in throughput and enhanced data quality for environmental single-particle genomics. Funding for the work came from the National Science Foundation, Simons Foundation, and the Research Council of Lithuania, with collaborators at Vilnius University and Atrandi Biosciences, alongside Bigelow Laboratory researchers. The authors see potential to extend the method to sediment and soil samples, expanding its use for studying environmental microbial communities and ocean viromes, especially elusive or hard-to-culture viruses. Beyond seawater, the approach could apply to sediments and soils, offering broad environmental utility for investigating complex microbial and viral communities. The research was conducted by the Bigelow Laboratory for Ocean Sciences and Atrandi Biosciences, using Gulf of Maine surface seawater, and the findings were published in Nature Microbiology around November 2025. Overall, environmental microcompartment genomics provides a rapid, scalable, and cost-effective platform for environmental viromics and microbiome studies, expanding the toolkit for exploring viral diversity in natural environments. The method yielded more complete and higher-quality viral genomes than some existing metagenomic approaches and captured viruses across the size spectrum, including Naomiviridae, an unusual DNA viral family. Unlike flow cytometry-based methods, this approach is size-agnostic and detects a broader range of viral sizes, revealing genomes that might be missed by other techniques. The approach produced more complete viral genomes and uncovered families like Naomiviridae that may infect abundant ocean bacteria, highlighting its potential to reveal previously hidden viral diversity. By enabling access to marine viral diversity and their hosts, the method addresses a major gap in understanding the ocean's microbial ecosystem, particularly the abundant but poorly characterized viral component. The Nature microbiology study showcases the first environmental application of this approach using Gulf of Maine surface seawater, demonstrating clear advantages over traditional single-cell genomics and metagenomics. Compared with traditional flow cytometry-based single-cell genomics, the method is size-agnostic, allowing sequencing of particles of all sizes, including large microbes, tiny viruses, and free-floating DNA. The technique uses thousands of tiny, semi-permeable microcompartments to isolate single particles in a minuscule water volume, enabling in-compartment DNA amplification, barcoding, pooling, and sequencing to reconstruct complete genomes without pre-sorting. In essence, thousands of tiny, semi-permeable bubbles compartmentalize samples for random single-cell or single-particle isolation, DNA amplification, barcoding, and sequencing to assemble genomes without flow cytometry. A new catalyst from the Korea Institute of Energy Research could reshape low-temperature CO2 conversion. Led by Dr. Ki-Young Koo, the team developed a copper-magnesium-iron mixed oxide catalyst that enables the reverse water-gas shift reaction at around 400 degrees Celsius, well below typical copper or nickel catalysts. The key is a layered double hydroxide structure that includes iron and magnesium to keep copper particles from agglomerating and to maintain activity at lower temperatures. This design fills the gaps between copper particles, sustaining stability and performance for extended operation. Unlike traditional copper catalysts, the surface directly converts CO2 to CO, reducing formate byproducts and maintaining high activity at 400 degrees Celsius. In tests under similar conditions, the catalyst shows higher formation rates and CO yields than noble metals like platinum, suggesting a favorable profile for low-temperature CO2 hydrogenation to CO and subsequent synthetic fuels. The reported CO yield at 400 degrees Celsius is 33.4%, with a formation rate of 223.7 micromoles per gram catalyst per second, and stability to over 100 hours. The work indicates potential industrial applications for producing feedstocks for sustainable fuels, including syngas for e-fuels and methanol. The findings were published in Applied Catalysis B, Environmental and Energy in May 2025 with DO1 10.1016/J.APCATB.2025.125475. The research aligns with KIER's broader effort to advance non-noble metal solutions for low-temperature CO2 hydrogenation and to support carbon neutrality goals. Dr. Koo emphasized the practical potential and plans to push toward real industrial applications using inexpensive metals. A new study maps the diversity of GABAergic neurons across the mouse telencephalon, linking developmental origins to adult circuits. Researchers used high-resolution whole-brain atlas data combined with prenatal and postnatal single-cell RNA sequencing to define a taxonomy that identifies 39 to 90 subclasses, totaling 284 supertypes and 1,051 clusters from over 611,000 cells. In the hippocampus and olfactory regions, long-range projecting GABAergic neurons in immature migrating populations create a variety of transitional and region-specific interneuron types, including CALB1 positive, CALR positive, and dopamine-releasing classes. In the striatum, medium spiny neurons resembling D1 and D2 types emerge as 7-LGE-derived subclasses, with subtypes that express DRD1, DRD3, or both, showing mixed identities and forming distinct patches across striatal and palatal borders. Two organizing principles emerge. Long-distance dispersion is common for telencephalic GABAergic neurons, and postnatal diversification varies by region, shaping local circuit function. The cortex is dominated by CTX-CGE and CTX-MGE GABAergic contributions, with roughly 10% of CTX-derived GABA neurons originating from the POA, including VIP, SNCG, LAMP5, ST, PVALB, and chandelier types, plus a newly defined RHPCOA-NDNF subclass enriched in hippocampal, olfactory, entorhinal, and CTXSP regions. GABAergic neurons arise from 5 subpallial domains, MGE, CGE, LGE, septum, and POA, and migrate to forebrain regions under the guidance of transcription factors and morphogens that shape fate and diversification. The study characterizes both long-range and local projection patterns, noting rare, broadly distributed PVALB-positive and STAT-positive subtypes and regionally restricted subtypes in the olfactory bulb, hippocampus, entorhinal cortex, and cortex, underscoring complex telencephalic integration. A newly defined MGE-derived LAMP5 LHX6 GABA subclass reassigns from CGE origins to MGE based on LHX6 expression, illustrating cross-regional presence of MGE-type interneurons in olfactory and cortical areas. In the cerebral nuclei, 4 major GABAergic neuron classes emerge in striatum and pallidum: CNUMGE-GABA, CNULGE-GABA, LSX-GABA, and CNUH-YA-GABA, with subtypes spanning dorsal and ventral striatal regions and palatal areas, including cholinergic and co-releasing GABA glutamate populations. The work delivers a comprehensive transcriptomic and spatial atlas of GABAergic neuron types across the mouse telencephalon, linking developmental origins and adult distribution to how interneuron diversity supports cortical and subcortical circuits. Overall, this study provides the most complete taxonomy to date of telencephalic GABAergic neurons, connecting adult cell identities to developmental origins and spatial distribution, and offering a framework for how inhibitory diversity underpins brain function across the mouse telencephalon. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.